Hello, in this video we will discuss about the protein digestion and nervous coordination. For example, the parasympathetic nervous system we will discuss in this video. So let's begin to understand from the digestive system. So the human is contain a digestive system with the oral cavity contain a salivary gland which produce the saliva for the lubrication and mastication through a teeth. So this is basically the parotid gland, maxillary gland and some sublingual gland which produce the amylase as well as the sodium bicarbonate etc for and produce the lysozyme for the bacterial um, resistance. So in this way here is you can see the esophagus, liver, gallbladder, duodenum, large intestine and small intestine and stomach. You can see in this diagram, and here is the rectum and anus. So let's begin to understand first of all the nervous stimulation we will discuss. So when the food will be checked through our vision, through our eyes, smell and taste, which that is detected by the higher brain cortex. This is the cerebral cortex. And after the detection, the after see and smell and taste, which that is give the mm, feedback, and this feedback is through our thalamus to the hypothalamus and ultimately the hypothalamus toward the medulla, and this toward the medulla will arise the parasympathetic impulse, parasympathetic nerve impulse along vagus cranial nerves number 10. So here is the protein, for example, which that is we will discuss in this video with the protein. So the cephalic phase is here. Cephalic phase means the detection through our brain. So that is known as receptor for sight, smell, taste, food, and activated thought of food and produce saliva and preparation for the food ingestion. So this is the part of ingestion after the swallowing of the bolus, and that will become a bolus. And this stimulate, and here you can see the thalamus will stimulate the hypothalamus, and the hypothalamus will stimulate the medulla, which contain a gustatory nucleus. And this gustatory nucleus are present in the medulla, which that is transmits the nerve impulse uh, toward the also the parasympathetic impulse along vagus nerves as well as toward the saliva production. So this is the salivary reflex. So this nerve is the vagus nerve, while on the other hand the smell is detected by our olfactory nerves and the gustatory nerves through our tongue, which that is used for the detection of taste. While here is a parasympathetic impulse along vagus nerve, which that is interconnected to the stomach walls, and in this way the stomach wall will stimulate to produce something due to the sympathetic nervous system will inhibit, while here is the parasympathetic nervous system which that is used for the stimulation of the digestion. So the digestion will begin in the stomach first from the oral cavity, and the second one is the stomach. So here is the second phase is the gastric phase. This is used for after the stretch receptor detect and distension of stomach, chemoreceptors detect increases the increase the pH, and the gastric phase will be occur. So increase the pH of the stomach due to the production of acid. That acid is known as the hydrochloric acid. So in this way here you can see this is the increased gastric acid and juice from chief cell, and that is the basically chief cell will produce the gastric juice, and the gastric acid is produced from the parietal cell. So remember about that. And the submucosal plexus which that is stimulated by this gastric phase, And this gastric phase is basically promote the digestion. And in this way, the parietal cells as well as the chief cell and enteroendocrine cell are also known as the goblet cell, which that is stimulate. First of all, we will discuss here is the amino acid, which that is the part of protein. This is contain a amino acid sequence is known as the peptide bond. CN bond is the peptide, which can be hydrolyzed to separate and to produce the free amino acid. So here is the chief cell and parietal cell and enteroendocrine cell. It is also known as the goblet cell or G cell. And in this way, here you can see this increased the gastric acid and juice from chief cells, parietal production, which inhibit the enteroendocrine cells. While the function of enteroendocrine cells to stimulate to produce the basically is the stimulate and produce the enteroendocrine cells, which produce the endocrine hormone, which stimulate the brain to relax and increase the parasympathetic nervous system. While the increase the gastric acid will inhibit this function. Why? Because the digestion will begin here. And here you can see the inhibition of the peristalsis due to the movement of the bolus in the form of chyme will be inhibited while the digestion will complete, so then it will be promoted. So the here is the increased gastric acid emptying, which that is promoted by the peristalsis. So this peristalsis, when the complete digestion will be occurred, so that things will be occur. So the next phase is the intestinal phase. This intestinal phase is stretch receptor detect distension of duodenum and chemoreceptor detect fatty acid and glucose in the duodenum. After the detection, so the next phase is the enteroendocrine cell, which produce the something we will discuss also. So on the other hand, the parietal cell and chief cell, which produce the pepsinogen, while the parietal cell produce the hydrochloric acid, which increase the acidity in the stomach. But the mucosal wall and the mucosal membrane will protect from the acid through autodigestion. So the ulcer will not occur if the mucosal wall or mucosal membrane is well grow. So here is the enteroendocrine cell, which that is stimulate after the intestinal phase to produce the CCK for bile production and the secretin, which that is used for the gastric juice and peristalsis decrease while the gastric juice will be increased. And the enterogastric peristaltic movement through a nervous system, which that is occur, and the juice reflex will be occur via a medulla, which that is as shown in the figure. So here is the pancreas. So let's begin to understand about the secretion of pancreas. But first of all, we should understand about the chief cell, which that is the mechanism of hydrochloric acid releasing into the lumen. So how it's possible? Let's begin to understand. So this is the our picture of the bigger picture of the chief cell, which contain a lumen inside in the stomach, while the inside is known as the inside is known as the parietal cell. So this parietal cell, not chief cell, here must be mistake, but you can understand that this is the parietal cell. So here is the hydrochloric acid produced. How? And here is basically chloride ion will move inside and the hydrogen bicarbonate will move out into the blood circulatory system. So in this way, the exchange of chloride, which that is move out through a chloride channel, while here is the water dissociate and this hydrolysis will occur and hydroxyl group will produce and proton. This hydroxyl group and proton will produce and the hydroxyl will bond with the carbon dioxide to produce the hydrogen bicarbonate. So while here is the anion exchange will be occur, which that is the same mechanism you can understand, and the carbon dioxide diffuse from the blood into the cell. So here is the again that's mechanism, and the carbon dioxide and hydroxyl ion will the carbonic anhydrase enzyme used to also produce the carbonic acid as well as the carbonic hydrogen bicarbonate. So this proton will be pumped out and the potassium will move inside. So this is the proton potassium ATPase pump. So here the potassium will again move inside and back and forth. You can see. So in this way here is the sodium will be moved inside by the exchanger of the proton, and this is known as the proton sodium exchanger. So this was the mechanism of hydrochloric acid releasing into the lumen. So let's talk about the hydrochloric acid effect to the pepsinogen first. So this hydrochloric acid will be produced by this mechanism. For example, the proton pump out and chloride also, and this will be bound together to form a hydrochloric acid. So this hydrochloric acid will be used for the activation of the pepsin. So this pepsin is released from the chief cell, and the chief cell release the pepsin in the form of pepsinogen in the form of exocytosis. After the exocytosis, basically the triggering through the basically the uh, secretin which that is used for the allow to trigger, and the gastric juice will be produced, and the peristalsis will be inhibit for digestion. So the complete digestion, after the digestion the peristalsis will begin, and in this way the peristalsis will inhibit the digestion basically, while during the digestion the peristalsis will be inhibited. Peristalsis means the movement of the food into the duodenum and throughout the digestive system through a contraction and relaxation of the circular and longitudinal muscle. So here is the hydrochloric acid, which that is basically chief cell produce the pepsinogen will activate by the hydrochloric acid as a substrate, and this is the removal of the um, pepsin half chain, and the pepsin will be formed, and this is the pepsin active, and this lead to digestion of protein. So this digestion is not completely, so that is known as the partially digested protein. This partially digested protein will form the chyme, and the chyme will move into the duodenum, and after the peristalsis, so it means the secretion from the gastric acid will be inhibited from the parietal cell and chief cell. So the secretion will be inhibited and the peristalsis will be occur. So here is the pancreas. We will just understand in the duodenum what happened, and the pancreas basically secretes the enzyme is known as the exocrine gland. This exocrine gland is in the pancreas we excrete the amylase and the amylase lipase and the pancreatic amylase and lipase, as well as the chymotrypsin. So this chymotrypsin, amylase and pancreas, which that is here, you can see the duodenum, which that is the cross section of the duodenal side. So this is the basically the brush border cell. It is also known as the enterocyte, which contain a brush border enzyme. So first of all, we will understand about the trypsinogen, which that is secreted from the pancreas, and after the secretion the enteropeptidases are present in the enterocytes brush border cell, which that is stimulate to the trypsinogen into the trypsin, and this trypsin is very important enzyme. This enzyme will be activate the chymotrypsinogen, proelastase and procarboxypeptidase, and this procarboxypeptidase, proelastase and chymotrypsinogen, which that is active due to the substrate of the trypsin enzyme, which will activate, and the chymotrypsin, elastase and carboxypeptidase will be formed. In this way the semi-digested protein will begin the further digestion. So this the long thread of the polypeptides is basically digested by this enzyme. After this digestion, the tripeptides, oligopeptides, and dipeptides and amino acid will be formed. This is the different types of the sequence of the amino acid in the form of tripeptide, oligopeptides, and the other things will be occur here. So here is the amino acid, dipeptides, and tripeptides to 9 peptides, nanopeptides, which that is known as the oligopeptide, while the more amino acid containing chain is known as the polypeptide. So these dipeptides, tripeptide, and oligopeptides will be further digested through a amino oligopeptidase and tripeptidase, dipeptidase, which that is all enzyme are present in the enterocyte plasma membrane of the enterocyte, and these will be digested further. And for example, amino oligopeptidases, which that is oligopeptides digest by this and to convert the dipeptides, amino acid and tripeptides. And in this way you can see in here this diagram which that is produce the amino acid and dipeptides and tripeptides. These tripeptides and dipeptides and tripeptides further digest through a other enzyme, the dipeptidase and tripeptidase, which that is all present in the enterocytes. So here I am not drawing that enzyme, but you can understood as well. So after this, the entry of the amino acid into the enterocyte and after this into the blood circulatory system is the mechanism, is the transportation of the amino acid. So how it's possible? Let's begin to understand. Here is the one proton will move inside and it is known as the simple transport of the amino acid. So here is the cytosol. After this, inside will move. Here is the amino acid transporter protein, which that is allowed to move inside into the blood circulatory system. So the sodium and proton exchange will be occur. In this way you can see. So here is the amino acid transporter is used for it, and this is basically the sodium will be move out, and in this way the sodium will again move inside through a ATPase, and this ATPase used for the potassium move inside into the enterocyte, while the sodium will move into the blood circulatory system, because the major cation is the sodium in extracellular while the potassium is the intracellular. So that is why. So here you can see this is the amino acid which that is move inside, and the one sodium as the basically the peptidase transporter protein. In this way the dipeptide will move inside and the cutting by the cytoplasmic hydrolyzation by the cytoplasmic peptidases, which that is cut and amino acid will be separate. Inside is also possible. So the dipeptides are also. So here is the proton and proton and sodium exchange will be occur. So I hope you make sense about the protein digestion, nervous coordination, and parasympathetic and sympathetic nervous system. So thanks.